Welcome everybody to the Harvard University Statistics 115-215, Bio 282, Biostatistics 282 class, Introduction to Computational Biology and Bioinformatics. I would like to congratulate everybody for your decision to take this class. In my study years, I just remember from both college and graduate school, even though I took many different classes, there were usually just a few that was really memorable and very useful for my later studies. And I certainly hope this course would be one of those, because bioinformatics is one of the most useful skills, whether you are working in statistics, computer science, and especially if you are a biologist. This is going to be a very, very useful skill. I would like to start the course with a brief overview of the history of bioinformatics. There are several waves of bioinformatics over the history. And the very first wave is the protein sequence and protein structure wave. In 1955, Fred Sanger developed the technology to sequence protein sequence. So he first sequenced the bovine insulin, which later enabled people to actually synthesize insulin as a drug. After the technique was invented, many groups use his technique to sequence proteins. And every sequence they obtain, they want to see whether other people have seen the same sequence or not. Therefore, they compare their new sequence with a previous protein sequence to see how similar it is. And so there are algorithms or computational algorithms that are needed in order to compare two sequences and see how similar they are. So this is called pairwise sequence alignment. So you look at two sequences, and if the two amino acids are the same, they're great, they're aligned well. And sometimes even if the amino acid sequence is different, you could see that they have similar biochemical properties, therefore could be aligned together. And so computational algorithms such as Needleman-Wunsch was developed in order to align the two sequences and provide a score to measure their similarity. And when I was a graduate student over 20 years ago, the first bioinformatics class started with learning Needleman-Wunsch algorithm. And so as people were sequencing more and more protein sequences, comparing one sequence with another using this type of pairwise sequence became kind of difficult. By the way, the reason people care about aligning the sequence, first of all, they want to know whether they have seen this before, but also because if two proteins have the same sequence, they might have the same structure, and therefore also they have similar functions. And so in 1973, a project was started for protein data bank. So at that time, some of the protein structures were solved. And over the years, all the solved protein structures were stored in this protein data bank. And if you go there, you can see not just protein sequence, but there are over 173,000 biological macromolecular structures available. And so they have the kind of the 3D structure of the protein. If we have two sequences that look similar, they might have similar structure, therefore they might have similar functions. And as we mentioned, if there are more and more sequences becoming available, doing the pairwise sequence alignment will become very expensive. Therefore, there are databases available to store all the publicly available sequence or newly sequenced protein sequences in a database. And there are algorithms for doing faster search. If you have a query sequence of your new protein sequence, you can enter into this database and run an algorithm called BLAST, which will tell you all the hits, previous sequences that might be similar to your query of interest. And so based on the sequence similarity, and if you look at what is known about these other proteins, you might be able to guess the function or the structure of your new sequence. And so a computational algorithm for doing fast search was developed. So instead of aligning sequence pairwise, it can very quickly search promising seed sequences and figure out which one is worth doing a pairwise sequence alignment. And so that's kind of a fast search algorithm. Also, around 1994, people started predicting protein structures. As we mentioned before, if two sequences have similar, if two proteins have similar sequences, they might have similar structure and also similar function. And it seems that once the protein sequence is known, usually the structure is set. And so over the years, people were using protein crystallography to solve the protein structure, but this is a very laborious process. And so there are computational approaches trying to predict the protein structure just purely based on sequence. Around early 1990s, many groups started working on this problem, and they might publish an algorithm to say, oh, my new method is the best. And they show some examples where they can do a really good job in predicting protein structure. But then, all these algorithms were claiming themselves to be the best. And so in 1994, this competition called critical assessment of structure prediction was established. Basically, this is a competition that runs every 2 years. In the first year, experts would contact protein crystallographer in the world to say, which protein have you just obtained the crystal? Because it could take years for people to obtain a crystal. Once they have a very pure crystal, usually in a year's time, from experience, they would know that the structure would be solved from crystallography. And so they would get a collection of proteins, usually in the 100-ish number. And then they would ask all the computational biologists in the world who are interested in this problem to make protein structure predictions. After the submissions are done, they would close the competition and then wait until a specific time. They contact all the protein crystallographers again to say, which of these do you have a structure already? They would use the experimentally solved protein structure as the gold standard to evaluate whether the computational predicted structure is correct. And so in this process, they can see which ones are the real winners, because instead of predicting one at a time, you would usually predict, maybe 50 or 100 at a time. And so this is a really unbiased approach. And also it's kind of blind ahead of time. Nobody knows the gold standard until they have submitted the prediction. Then they can look at the results. So over the years, actually a lot of computational biologists who thought, oh, protein structure prediction was a really easy problem. They came in and, after a few rounds of this CASP competition, they dropped out of the whole field completely. And so, but then there are people who learn from their mistakes, they continue to improve their predictions, and there were kind of winners over the years. We'll show you some of the recent results later. One thing people were trying to use in order to predict the protein structure is to see whether in the protein there are different functional domains. For example, these are families of proteins. You can see here, they contain small stretches of sequence that look very similar. Usually they have similar sequence and therefore small functional domains and in similar structure and similar function. It's as if you say any kitchen would have a counter, would have a refrigerator, would have a stove and a sink and a microwave, right? So, if this protein is gonna have a function like this, they would need these different domains in place. And so a lot of times when people are trying to predict protein structure, they first try to locate these domains, and based on this, they can predict the structures better. So around the same time of CASP competition, a blocks database was established, which identify these protein domains, which are very important for predicting protein function. And so continuing on the protein structure prediction, we can see this is CASP9 in 2012. So usually after a competition, the experts will evaluate all the predictions and they will write a summary paper to be published. And so you can see here that year, the columns here includes all the structures that were solved, and they can see which algorithm predicted well or badly. And so each dot here is one prediction, and the target or the proteins that are easy to predict is on the left, whereas those that are really hard to predict are on the right. And so each target, you can see there are multiple predictions. And here shown in two different colors are the kind of the overall best two predictors. One is HH-Pred, the other is Rosetta. And you can see in the easy target, they can predict. And so usually the measurement of whether something is predicted well or not is called global distance test, which kind of evaluate, the agreements between the prediction and the truth or the native proteins that are solved from experimental approaches. And so you can see when the global distance test give you a score above 90%, the correct structure, which is drawn in red in here, versus the predicted structure, have highly similar, you can see the structure aligned very, very well. So this is easy to predict, and at least the prediction and the correct structure are very, very similar. Whereas as you can see towards this end, when you have a structure that is really, really hard to predict here, the global distance tests only have a score of 10 or 20. And you can see here the correct structure and the predicted structure still look quite different. So this is the kind of state of the art in 2012. In 2018, a new player came into the field. This is from DeepMind, it's a Google company using AI to make predictions. They just use the existing PDB database to say, if I have the sequence and I have the structure, can I learn the rules? And so a lot of the other computational predictors at the time, they have to use years and years of biochemistry and protein structure knowledge to create the rules, whereas AlphaFold is kind of trying to learn the rules from just all the existing sequence and structure. And they joined the competition in 2018 and was a very clear winner in this competition and beat the second player by a lot. And you can see at that time, among all the predictions that it made, the average global distance test of all the structure it predicted is about 60, which was already much, much better than the second player. But what is even more amazing is by 2020, 2 years later, in AlphaFold 2, this is CASP14, the average prediction can reach about 90. Actually, a score above 90 is considered roughly equivalent to the experimentally determined structure, which a lot of people in the field consider that protein structure prediction problem on a single protein is a solved problem with AI, which is really fantastic. Of course, there are still new problems, such as when you have a big molecule which includes multiple protein in a complex. How do you really predict that this big complex, and how do you predict protein-protein interactions? So those are still unsolved problems, but we are hoping, really with computational biology or AI, that over time you can get better and better at this. The second wave of bioinformatics is the gene expression wave. In 1977, a technique called Northern blot was invented in order to measure the expression of a single gene in a cell condition. This is an experiment that also took a lot of effort to do. In 1995, new technology was developed at Stanford, which used microarrays to measure hundreds or thousands of genes at a time. I remember my undergraduate research advisor came back from a conference in 1996, and he was so excited. Basically, you can use a cellulose membrane and have hundreds of tiny dots or probes, each probe measuring the expression of a single gene. Therefore, using this microarray, you can measure many genes in a specific condition. And so in this experiment, usually you have cells grown in different conditions, or samples taken from different tissues or different conditions. For in this case, it's normal cell versus cancer cell. You isolate the RNA and fluorescently label the RNA, or convert them into cDNA, fluorescently label the cDNA from two different conditions into different fluorescent colors. And then you mix them together and hybridize this onto kind of this, initially was a cellulose membrane, later on was a glass slide. And the glass slide have these little probes. Each probe will measure the expression of one gene. And you can see the RNA for the gene in the two different samples will be hybridized to this. And then after hybridization and wash, you shine color onto the slide. You'll be able to see based on the color for whether some gene is higher in the normal condition or the cancer condition. For example, if it's dark, it means that this gene is not expressed in either conditions or expressed pretty lowly. Whereas if it's yellow, this gene is highly expressed at similar level, but in both conditions. But if it's green, this is a gene that is highly expressed in the normal cell, whereas if it's blue, it will be a gene that's highly expressed in the cancer cells. And so this is really fantastic. People can really use it to measure expressions of hundreds or thousands of genes at a time. And so many groups started adopting these type of microarrays to do expression studies of the condition they are interested in. Around the same time, there were also commercial solutions. So initial cDNA array was generated in a lab. The PhD student or postdoc would create those spotted cDNA arrays, but between conditions, sometimes you have very significant artifacts. And so there were industry solutions. And so in this array, there could be 6 million probes measuring all 20,000 genes. And it's because it's industrial manufacturing, the results were much more reproducible. And so people were, these are kind of more expensive, but people started using this for measuring expression. And I remember this is a 1999 study, seminal study done by Todd Golub at the Dana-Farber Cancer Institute, where I'm a faculty. So basically, there were two types of leukemia. One is a acute lymphoblastic leukemia. This is called ALL. The other is AML, acute myeloid leukemia. These two types of leukemia pathologically look quite similar. So sometimes it's hard to distinguish them for diagnosis. However, the treatment and outcome could be different. Therefore, reaching a correct diagnosis is quite important for treatment and for the patient. And so in this study, Todd Golub and his colleagues collected a number of ALL patients samples and AML patient samples. So each column is one patient sample. And then they profiled RNA on this commercial microarray to look at the expression of thousands of genes in each sample. And by looking at the differentially expressed genes, they were able to identify gene markers, for example, that are much higher expressed in ALL compared to AML. And they can also identify genes that are highly expressed in AML. So the goal is that in the future, if you have a new patient, from pathology, maybe the doctor is having a difficult time telling these two cancers apart, you could profile the patient tumor or the blood sample with the microarray, and based on the expression of these marker genes, really reach a much more accurate diagnosis and give the patient the right treatment. And so this really kick off precision medicine. People are doing a lot more expression profiling experiment. And so if you look at gene expression in 2020, there are assays such as RASO-seq or Luminex assay. Basically, these are technologies that can profile the expression of about 1,000 important marker genes at very, very low cost. When the first Affymetrix microarray were invented, it cost close to $2,000. Then it dropped to $1,000 and then to like a few $100s. But now you can see here with RASO-seq or Luminex, you can do this with a few dollars per sample. And so with this type of technology, for example, recently the Broad Institute has a connectivity map project where they use this Luminex approach to profile over around a million different profiles of many different cancer cell types being perturbed in different conditions by either gene perturbation or a drug, a treatment. And then they can measure the expression of 1,000 genes. After they get the data, they can use the publicly available gene expression profile where people measure the expression of all 20,000 genes, and then use the 1,000 genes they measured to computationally impute the remaining expression. And then they can get really kind of a genome-wide expression signature of how the cell would respond to a particular perturbation. So these are really very useful resources for people to look at drug or gene effect. In addition, people are now developing single cell gene expression techniques. So instead of looking at a bulk tissue, single cell expression technique can look at within a complex tissue whether there are different cell types. For example, in the brain, there are different components, and also in the heart and in liver, there are so many different cell types. And so this is a recent study that was published last year in Jay Shenduri's lab at University of Washington, where they took 15 different fetal organs and 121 different samples, and using a technique that they developed called siRNA-seq. It's a kind of a split pool approach, which we will discuss later in this course. And using this approach, they were able to sequence over 4 million single cells. It's really mind-boggling. It's over 4 million cells. Each cell, they can reach hundreds and thousands of genes. So it's like a tremendous data matrix. And based on that data, they can cluster the cell types and also cluster the different tissues and organs, and then figure out how different type of cells in different organs are expressed and how they interact with each other in development. And so I hope you can see that the technology for gene expression also have evolved a lot, and with it there are a lot more data generated, and also computational biology is really essential for making sense out of that data. The next wave of bioinformatics is the DNA sequencing wave. If you look at the previous slide in here, we mentioned single-cell RNA sequencing. This is actually a DNA sequencing approach, right? So in 1953, Watson and Crick discovered the DNA structure to be a double helix. That's kind of the beginning of molecular biology in some sense. And in 1972, a technique was developed called recombinant DNA. It's an experimental approach that allows people to cut DNA and stitch DNA together to create new pieces of DNA sequence. And so this initially, people could do this type of experiment in the lab to cut and stitch and create new pieces of DNA. This is kind of, many groups started using this approach to study the gene they're interested in, to look at functions. It was very exciting time. However, at that time, sequencing DNA was still very, very difficult. So this is a study that was published in 1972 with a single author. Actually was the PhD thesis of Jerry Rubin, who is now a professor at the Howard Hughes Medical Institute. And if you read the abstract of this study, it says the nucleotide sequence of yeast 5S ribosomal RNA has been determined to be this particular sequence. And this whole paper basically is about 5 years of experimental work to determine the sequence of one gene. And actually this now only takes 1 second to do. So this is because over the years, there are better and better techniques developed for DNA sequencing. So for example, in 1977, Fred Sanger, after having invented the techniques for sequencing the protein sequence, now also developed a technique to identify the DNA sequence of a DNA. And so at the time, the DNA liquid sequence were limited because you have to have enough amount of DNA for the same thing to sequence it. So until about 1985, the polymerase chain reaction was developed, which allow people to amplify any specific piece of DNA many copies. This is kind of an exponential amplification, starting from a single copy even, you can amplify millions of copies in a very short time. And with enough copies, then you can use Sanger sequencing to really figure out the specific sequence of some gene you're interested in. And so with that, more and more people are starting to do DNA sequencing. And we mentioned previously, the National Center of Biotechnology and Information were started in around 1988. So this, if you go to the main NCBI website, this is from the National Institute of Health. There are many useful resources here. From the software, you can see the BLAST algorithm for looking at query. If you have a query sequence, you can compare this database, whether it's a protein or DNA, it will tell you whether other sequences look like your query sequence. There are some information on protein domains and structure, on genes and gene expression, on genetics and medicine, on whole genome sequences and mapping of genome sequences, on homologies between the same gene in human versus mouse versus rat and different species. On literature, I think a lot of biologists probably know PubMed. It has like millions of published literatures. This is also protein sequence analogy and also taxonomy, basically how terms, disease terms or molecular biology terms are linked to each other. And there are a lot of useful training and tutorial. And so at that time, more and more people are looking at genome sequencing. I remember when I was an undergraduate, we used to use Sanger sequencing to sequence at that time a bacteria a parasite genome. And we would sequence it out and then check using BLAST to see whether this piece of DNA has been sequenced before. And if it has been, we will discard the sequence. But if it's a new piece of sequence, we cheer, we submit this into NCBI as a newly sequenced DNA for that new parasite genome. And so the BLAST, again, was kind of the algorithm we all used computationally to check the sequence against our query. And so around 1990, NIH started a project for the Human Genome Project. So because people felt that the technology, with PCR, with Sanger sequencing, the technology for sequencing the human genome is a possibility. We just needed the will and the money to make it happen. And so Francis Collins, who is currently the head of the National Institute of Health at the time, led the National Human Genome Institute to start this project. At the time, there were hundreds of scientists, including US and 6 other countries, joining the efforts. And their approach for sequencing the human genome is a divide and conquer approach, or clone by clone approach. If you think a human has 23 pairs of chromosomes, it's like an encyclopedia. There's a chromosome 1 is like your encyclopedia on letter A. Chromosome 2 is like a encyclopedia on letter B, and so on. And so initially, a lot of the work were about dividing the genome into chromosomes, roughly figuring out where things are in each chromosome, divide the chromosome into chapters and into mini chapters, and then go to a particular page in the encyclopedia, then shred the page into small pieces, and then sequence them out. Usually this has to be done repeatedly so that there are sequence overlaps for this page. The sequence similarity allow people to align the sequence together at the end to assemble the sequence into something called a contig. And then the contigs are gradually stitched together to create that chapter for one chromosome. And so this is an extremely laborious process. In 1990, it was estimated to be finished in 15 years. But in 1997, there was a private effort led by Craig Venter in a company called Celera. They said we could do this much, much faster. Instead of dividing the whole human genome into chromosomes, into bigger chunks and then smaller chunks and then shred it, the private effort was using a whole genome shotgun approach. Basically, this whole encyclopedia is shredded into all the smaller pieces and sequenced many times over. And purely using computational approaches, looking at the similarities, this approach could assemble the genome into contigs and into whole chromosome sequences. So this actually created a lot of pressure to the public effort, because the private effort eliminated the really time-consuming and laborious process of dividing this genome into smaller chunks and sending it to different genome centers. But at the time, the private effort also claimed that they could do it faster and cheaper, which at the time, the public effort initially was kind of skeptical. But because the competition from the private side, it really motivated both sides to accelerate their technology development, automation. So at the time, there were better sequencers developed. And so the technology improvement allowed people to sequence large amount of sequences much faster. And this computation from Celera also stimulated the public effort to use more pipelines and automations to speed up the process. And so because of this competition, eventually the White House have to be involved. They work with both groups to say, let's try to work together. And so they agreed that, there's kind of a public announcement. The public effort and the private effort actually finished at the same time in the spring of 2000. And so the private effort at the time, you can imagine this is actually a massive computational undertaking, having to stitch together so many sequences to create the genome sequence. But even in the public effort, at the time, they just said, okay, let's accelerate the process. They also started doing a lot of sequencing. And at the end, it was David Hausler and Jim Kent from the UC Santa Cruz. They developed also an assembly approach to assemble the public effort, from the pages to the small chapters, to the big chapters, to the whole book together. And so it was actually printed and still warm when the news announcement came around that, the public and private finished the draft sequence of the human genome at the same time. And the complete human genome was finished in 2003. Of course, this is not really the end. This is a reference genome. I think at the time of the draft genome, it was 9 people's DNA mixed together, which only kind of represents the common sequence shared by those individuals. Over the years, people have found that, between individuals, we have still some differences. So now people are sequencing your personal genome and her personal genome. So this is kind of a additional problem. And so you can see here, with the genome sequencing, there's a lot of work on the assembly, and later on also computational approaches for predicting genes and also understanding what the genome sequences do. If you have a difference between one person to another, does this give the person blue eyes or blonde hair and things like that? And so these a lot are based on an integration of experimental techniques, generating a lot of data, and computational approaches to analyze the data. So I hope that you can see the Human Genome Race, again, is a project where computational biology really played an essential role to the success of these projects. Throughout this whole course, we are going to teach you how to do sequence analysis, high throughput sequence analysis. So let's revisit high throughput or sequencing. In the beginning of 2001, this is right after the human genome sequencing was finished, or the draft genome was sequenced. If you look at a typical genome center, these are actually quite big genome centers. Usually you have a huge room like this of equipment and about 35 scientists working together to prepare a sample. In 3 to 4 weeks, they will be able to prepare enough samples to be put onto the sequencing machine. Then there is another room, also very big, with 74 of these capillary sequencers. And this is the sequencer that I was kind of using when I was in college. And usually these 74 machines will be run by 10 people, and a big genome center can run about 15 to 40 runs per day. And they can generate about, each of this big sequencer can generate 1 to 2 megabytes of sequence per day at a total capacity of about 120 mega per day. And that's kind of the workhorse for both the public and private efforts to finish the human genome project. So for many years after that, people were using that approach to sequence monkey genome or dog genome, more other species. But then in 2007, a technique was invented called high throughput sequencing, or second generation or next generation sequencing. This is different in that we don't sequence one sequence at a time. We can sequence millions of sequences at the same time. And so for the previous Genome Center now can be really streamlined to a process like this. So with this one machine, one scientist could prepare the samples in a day for the DNA to be sequenced. And this, for sequencing, you buy this machine. This is actually early days, a genome analyzer. This is from the same scientist. After finishing the sample preparation, can load the DNA sample onto this glass slide. You can see here, this is the size of a penny. So it's a very small slide. It can, you can load millions and millions of sequences onto this glass slide. And you put this in this machine and start the run, and just kind of once in a while come and check the run. And so usually in 3 to 5 days, the run will finish, and this instrument can really output 0.5 gigabyte of data per day per instrument, which is, compared to 2001, it's an amazing progress in the technology. And Illumina was the market leader in this. And if you look at genome sequencing now, or sequencing technology now, Illumina is still the market leader. And I love to present this every year to look at the updates from Illumina website. And so they now have a suite of high throughput sequencing machines. There are the mini ones, which usually individual labs can buy. It doesn't generate as much sequence, but still it's quite impressive. And so each run you can see can take a few hours to a day, which can generate gigabytes of data, and in millions, like 4 to 20 millions of reads. Each read is usually 150 base pair long, and you can have bigger machines which you can run from small core facilities here, which runs for 2 days or 3 days, and it can generate hundreds gigabyte of data and up to 1 billion reads per run at this read length. And at bigger core facilities or companies that run sequencing as a service. So sometimes a lab would generate sequencing samples, and then they send it to an outside company to be sequenced. And these NovaSeq 6000, you can see these machines can generate up to 6 terabytes of data and 20 billion reads in less than 2 days. Even thinking about this data and like where to store and how to transfer this data is kind of mind-boggling, right? Compared to how much people were sequencing in 2001, you can imagine this is just one machine. And very often, big genome centers or sequencing companies might have 20 of these machines, and many universities also have one of these machines in their big core facility. And so the data that's generated, it's humongous. And so if you look at the cost of sequencing a human genome, this is 2001 when the first human genome was finished. At that time, sequencing one human genome would cost $100 million. It's actually the full human genome project at the time cost $3 billion. But because the technology was improving, by the time 2001, sequencing that same genome would have cost 100 million. And initially, this was kind of a small price decrease, but it was kind of decreasing because the technology was improving until about 2007, when high throughput sequencing came around. Instead of sequencing one DNA stretch at a time, you can sequence millions at a time. And the cost suddenly started to decrease much, much faster. It's even faster than Moore's law. So if you look at this line, this is the Moore's law in the semiconductor industry. This was used to develop to describe the silicon chips that were used in computers, which dictates that every 18 months, the capacity of the chip would double and the price would be half of before. And so initially, with first generation sequencing, we were following the Moore's law, but with the high throughput sequencing, the price of sequencing a genome decreased really, really quickly. I remember in 2007, 2008, one of my colleagues was asking me, he said, oh, I have a really rich friend and he wants to sequence his genome. Should he do this? And you can see here at that time, it would cost about $1 million to sequence his genome. I'm like, nah, don't do it. It's too expensive. And at that time, even if they were to sequence their genome, the information on what they would learn is still kind of questionable. But you can see here, nowadays, sequencing a genome costs less than $1,000. Yeah, so now this is a reality. If you want to sequence your genome, you can do it. It's not too expensive, right? And also because so many genomes are sequenced, now people are learning a lot more about this. But first of all, with all the data generated, we have to assemble the genome, identify the difference between this person's genome with that reference genome to see which positions are different. And through this, you can then try to figure out whether this person have a different trait. And so to do the assembly and also identify the differences, I want to show you one algorithm that was published in 2018, where scientists were using deep neural networks to identify SNP or indels, which are kind of small nucleotide or a few nucleotide differences between individuals. And these SNPs or indels happen usually occur 1 in every 1,000 bases between every pair of human. But then they could create quite different traits, such as intelligence, height, hair color, or nose shape, or whatever, like many different traits. And so this actually have very important implications. When people have sequenced enough genomes and look at the genome sequence, they can say, aha, this piece of DNA is important for hair color. This particular region is really important for a perfect pitch in music, or intelligence, or height, or things like that. And also, probably more important, if you can identify a specific region of the genome which is responsible for a disease susceptibility, such as likelihood to develop cancer. So we probably know that Angelina Jolie carries such mutation, or a specific DNA in her genome, which made her more susceptible to breast cancer. And so she decided, after she had kids, to have a preventative surgery to remove her breast. This would drastically reduce her risk of developing breast cancer. And so you can see here, based on the genome sequencing, you could learn, whether you are likely to develop a different disease. And based on this knowledge, you could pick a better lifestyle or have preventative procedures to reduce the risk for disease. There are also high throughput approaches to do disease diagnosis and also for drug treatment. I want to mention this, which is really already happening in major cancer hospitals. For example, at the Dana-Farber, if a patient is diagnosed with cancer, their tumor samples will be sequenced. We don't sequence their whole genome. Usually we just sequence about a few hundred genes that over the years have been discovered to be frequently mutated. And there are also drugs that can potentially be used in order to treat cancers that carry these mutations. And so these are called targeted therapies, which means that they only kill the cancer cells which carry the mutation, but will leave the normal cell intact, which is quite fantastic because it would have less side effect than, say, chemotherapy, which will just kill even your normal cells and have a lot of bad side effects. Yeah, so basically now every patient coming in will get their first tumor sequence done. At Dana-Farber, this is actually done with philanthropy for free to the patient. And based on the tumor mutation profile, very often this patient would be given a specific drug based on that mutation to kill their cancer cells specifically. And sometimes this patient will also be matched to a clinical trial to be tested for a new drug, which might target one of these mutations. And so you can see here, with the cost of sequencing genomes reducing so quickly, you can then really have personalized disease prevention, diagnosis, and also personalized medicine or precision medicine to treat the disease based on the specific DNA sequence. Of course, this again generates huge amount of data, and we are faced with big data challenges. And that's kind of a big motivation for biologists to learn more computational biology, and also it gives a very interesting opportunity for computer scientists, for statisticians, for physicists, for engineers to really develop computational solutions to solve this big data challenge in biology and medicine. This course is an introduction to computational biology and bioinformatics. So you might wonder, what is the difference between these two terms? Bioinformatics is focused on the creation of tools such as algorithms, databases, web servers that solve problems for biologists. The goal is to build useful tools that can work on biological data. It's more focused on the engineering aspect or the quantitative aspect of the problem. Whereas computational biology is focused on the study of biology using computational techniques. The goal is to learn new biology knowledge about the living systems, understanding the mechanism. It's more about the biological discovery. And so you can see these two are slightly different. In this course, though, we're going to use these two terms interchangeably. A few years ago, I wrote a blog to look at the different levels of bioinformatics or computational biology. I want to quickly kind of go over this. The level 0 is something I found in the early days. I remember going to a conference, some people ask, I think it was like a panel discussion. They ask, there are already a lot of public data available. What should we do? And so I asked them, well, it depends on what questions you are interested in solving, what problems you are interested in. And they say, we're interested in modeling problems. And so we know that actually in this field, there are people who are doing modeling for modeling's sake. They publish papers that, you know, neither computational people or biologists care about. I hope that if you want to learn computational biology, you don't go to there. This is kind of not the right direction to go. Instead, for this course, and I think most of the computational biologists, when they are starting to learn bioinformatics or computational biology, they start with level 1. You can understand some, basic biology, look at some real public data, use published tools to analyze the data and generate hypotheses for experimentalists. For example, You might be within an experimental lab, or it could be your own data, or you might be collaborating with an experimental lab and they have generated the data. Initially, when you are learning, use that and try to use existing tools. Maybe you will write a small script here and there, convert the data into the right format before you can run some published tools, and just kind of help QC the data, make sure everything is working, and generate some hypotheses. And so this is the entry level. You just use published tools to help some experimental collaborator. In this course, this is where we're going to start. But we are going to teach you a lot of algorithms that were developed under level 2. So these work are focused on developing algorithms, database, web servers for analysis. There is a need for such algorithm, especially in the beginning of the technology development. For example, when there is a new sequencing technique like share-seq or multi-seq or crop-seq, there are so many different new something-seq techniques developed. Usually when these techniques were developed, they are not very good accompanying computational solutions. And so many labs that were generating or adopting these technologies may not even know whether their experiment worked or not. And so many computational biologists would develop an algorithm to help the experimental biologists, once they generate the data, to analyze it, QC, and generate hits. And so the difference between the level 1 and level 2 is, level 1, because you are using published data, you can only help your collaborator, whereas level 2, once you have the algorithm, many groups in the public can use your published algorithms and analyze their own data. And when a technique becomes more popular, there are more and more public data generated. Usually there will also be need in order to reuse publicly available data and integrate them together to make discoveries. And so that leads to level 3 of bioinformatics or computational biology, which is you can either use public data or some of your private data, and also computational approach wise, either you use your own algorithm, or you can use public algorithm, but in a very unique or smart angle to actually make discoveries. And so now in the entry level, the computational biologist usually is only having a supporting role, whereas in the level 3, the computational biologist now is taking the front and center seat. They decide, I'm interested in using public data to answer a biological question, to make a discovery, and I'm going to use all the computational tools and public data at my hands to make that discovery. And so usually this would take integration and modeling, but then the goal is really to make discoveries. Over the last 20 years, because of the technology development, there are also many consortia from US and other countries where a group of scientists will form a team. They might be coming from different institutions and they would work on a big project together. For example, generating the expression data for thousands of tumors, or in the brain, look at the expression of all the different cells, or look at the epigenetics of many different cell lines together. So usually these consortium efforts generate a lot of data, not just for the data generators, but also for the community to be reused. And also there are usually very good computational scientists in those teams to help integrate the studies together for publication. And so if you are a computational biologist, you start with level 1 to kind of help other people analyze their data. Maybe they already have hypotheses, you have a supporting role, and then say during your graduate school years, you learn how to develop algorithms, and then towards the end of your PhD or beginning of your postdocs, kind of learn how to do level 3, which is you have the problem, you own the problem, using the public data to make the discoveries by integrating public data using good integration and modeling approaches. And also, if you have an opportunity, participate in this consortium project to see how team science is done. And so these are all very useful endeavors from 1 to X. So bioinformatics and computational biology is a very interdisciplinary field, and it has attracted mathematicians, statisticians, computer scientists, engineers, biologists alike. In fact, the emphasis of the importance actually change over time. We find that when there is a new technique, especially now with sequencing, the early winners are the computer scientists, because they can write program fast. They can get all the low-hanging fruits, automation, parallel processing. So these are done very quickly. That's at the sequence level. With high throughput sequencing, at least you have to know where the sequence come from, map it into the genome. But then once you have done this, you look at many different samples and you say, what are the difference between this drug condition and without the drug condition? And you would be looking at numbers now. This gene is expressed at this level in one condition, but expressed in a different level in a different condition. Once you look at numbers, then the mathematicians or statisticians usually will play a bigger role. And they will develop normalization methods, how to robustly identify differential levels in the data. And then after these statistical questions are solved, and then later on in the integration, there are additional, of course, both engineering and statistical challenges, but the bigger question is what biological question we're interested in answering. And so you have to really look at enough public data, use solid CS and statistics or machine learning, but then really answer a good solid biological question. And so you can see the emphasis really change over time. This is a very applied field. Over the years, I have seen from freshman undergraduate students to postdocs taking the class, and not always undergraduate student would do worse than graduate student. And so we would really welcome everybody who might be interested. This would be very, very useful training for many, especially if you want to be a biologist or maybe a doctor in the long run. And the more you practice, the better you will get. We're hoping that this course will bring you into the door, but you will continue to practice and use it and become an expert over the years. The field really moves quickly. It also moves with technology development. As you can see, in the early days, people were doing protein structure prediction. In fact, when I started my PhD, my advisor wanted me to do protein structure prediction work. But then microarray was just invented at Stanford. I was a PhD student at Stanford. Many other senior students and postdocs and faculty were telling me, microarray is new. It's time to kind of explore this new direction, which will give you a lot more opportunities. And so that's what I did. And so recently, you can see in this course, for many years, we covered microarrays. But last year, we decided to take that out, and we added more coverage on single-cell RNA-seq and single-cell ATAC-seq. And so even though not all the old problems are solved, the new techniques, because they're so powerful, they create a lot more computational challenges as well. And so the scientists would just kind of move on to the next exciting questions. You might wonder whether this class is for you. We do look for students who have computer programming. Yeah, usually R and Python programs are the ones we're going to use for this course. We also hope that you have basic molecular biology and genomics background, because we will be covering a lot of biology, especially on gene expression, epigenetics, gene regulation, GWAS studies, and also cancer genomics and cancer immunology. So I think knowing some good molecular biology would definitely be helpful. And also we need students to have some basic statistics. For example, do you know how to do hypothesis testing, to understand different distributions, intuition between those distribution and statistical tests? And so these are some of the requirements in the course. So in this class, we are going to cover the major questions and major solutions in computational biology, especially those that are related to gene regulation, whether in normal physiological situations or diseases. We will cover the intuition behind many of the algorithms behind it, but we're not going to ask students to develop those algorithms. That's really too hard. Instead, we will teach you hands-on skills to at least play with your data, maybe write some simple codes to manipulate your data, and then say convert your data into a certain format so that you can run some published algorithms. So we'll teach you a lot of hands-on skills in data analysis and running algorithms. So hopefully that you can learn how to troubleshoot, how to understand the README, how to run a new program that you have never tried before, and how to look at the results. This is not a course for statistical theory or deductions. And so if that's what you are looking for, this is really the wrong course for you. We will be covering different kind of major biological areas, the genomic techniques that were used there, the different data types that are generated, the different computational algorithms that are used to make sense out of that data and show you how to run it. That's kind of what this course will teach you. If you want to see whether your biology, statistics, or computer skills, whether it's sufficient, I would encourage you to look at homework 1, which we have already released, which will be due in 2 weeks' time. No matter how long it takes you, the remaining homework 2 to 6 will take at least twice as long. And so homework 1 will really give you a sense, are you prepared? Can you really handle it? I would argue though, that if you want to study medicine or biology, computational biology is going to be one of the most useful skills you're going to have. And for that, I'm going to quote Sidney Brenner. So he is a Nobel laureate in 2002. So he won the Nobel Prize for identifying every individual cell in the C. elegans worm. And so basically from one in the embryonic stem cell, how this cell goes through, basically lineage tracing, figure out, all the 900 cells, where they go later. And so he won the Nobel Prize in that. And this is a biologist who did experiments throughout his life. And in a 2008 interview, he said that we presently have huge amounts of data that are largely unassimilated. I have discovered that while there are rewards for collecting data and distributing data, there is little, if any, support for organizing it. It is what I call low input, high throughput, no output science. I think this really says, emphasize the importance of computational biology and bioinformatics. It's really essential in understanding the data. By the way, low input, high throughput, and no output science is something, say, a scientist before doing an experiment are not really thinking. They just say, oh, I have this interesting sample, let's just throw it into the sequencer and see what we get. So they generate a lot of data, and then they also don't know how to do computational analysis. So at the end, it's really a data paper with no real findings. And so there's no output science. And so I would say computational biology is really, really important, because right now, new experimental techniques are being invented all the time. They are so powerful in discovery, and yet they generate a lot of data. And so before designing an experiment, think carefully what this technique will really give me, how to really design an experiment. But then once you get the data, either learn the computational analysis yourself or collaborate with a computational biologist so that you can make sense out of your data and really answer some important biological questions, and think hard, and have the necessary skills to design a cost-effective experiment and really reach some real insights. Because of this, in 2017, another colleague of mine, Florian Markowitz from University of Cambridge, published another paper called All Biology is Computational Biology. As we mentioned, so many powerful techniques now generate high throughput data. It's not uncommon for an experimental biologist to spend a few months to learn a new technique, to do the experiment well, to generate robust results. However, if they don't know computational biology, they could spend even more time in front of a computer, staring at a screen, trying to understand whether their experiment worked, what result was actually generated, what's the important hits, what's the insights that's coming from their own data. And very often the speed of the analysis and also the effectiveness of the experimental computational integration is key to the discovery and also the eventual publication of the study. If you don't know how to analyze the data, you can be totally scooped by another group that did the exact same experiment but can analyze their data faster. And so for an experimental biologist or somebody who is going to medical school, you should ask yourself, can I afford not to learn computational biology? This course is hard. You will spend a lot of time doing the homework and learning the different algorithm and troubleshooting. But think about the months and months of work spent to learn one molecular or genetics experimental technique. You still have to also work day and night to learn as well. And so for this course, I would say bite the bullet, learn it well. If you can do it well in this course, you will never be afraid of big data in biology in the future. So some information about me, I'm Shirley Liu. I'm a professor of biostatistics and computational biology at the Department of Data Science at Dana-Farber Cancer Institute. I'm also a professor at the Harvard School of Public Health and also in statistics department at Harvard University. That's why this course has a Stat 115-215 title. So my research is on computational biology in translational cancer research. We work on gene regulation, transcription epigenetic gene regulation at the bulk level or single cell level. We also use CRISPR screens to look at gene regulation. And recently, we are heavily invested in understanding cancer immunology. So if you want to know more information, you can go to the lab website. We'll also post a video about our introduction. And so this year, because of COVID, both FAS and Harvard School of Public Health were able to allocate more TAs to make sure that students who are taking the course remote will have enough help. So as a result, we have 6 TAs sign up for this course, and they will be hosting the labs for every week and also on the weekend office hours in order to answer questions you have regarding the labs and also help you with homework. Okay, so I'm sure you will be working a lot with them. There are some information on the course organization. This year, we're going to divide the course into 4 different modules. The first module is on RNA sequence analysis and sample classification. You will learn read mapping, how to do differential gene expression, how to cluster the samples, look at the gene annotation of a group of genes, or and also remove batch effect. Sorry, there's a typo here. And also how to do dimension reduction to cluster the samples, and also learn supervised machine learning to classify samples, for example, to see whether a new sample is normal or diseased for diagnosis. In module 2, we will cover transcriptional and epigenetic gene regulation. So, basically module 1 will show you these genes are differentially expressed, but then the second module will tell you why they are differentially expressed, right? So here we cover transcription factor motif finding, ChIP-seq, DNA methylation, histone modification, how to use hidden Markov models to annotate genome segments, and chromatin accessibility and higher order chromatin interaction, and how to use these type of data to understand gene regulation. The third module, we will cover GWAS interpretation and single cell data analysis. There are courses, a whole class, a semester course to cover GWAS. So we are only going to use 2 lectures to cover this. So we'll make them really brief to kind of, basic information to give you basic idea of what is a GWAS study, what is SNPs, LDs, what are some of the public resources to evaluate GWAS, and how people use functional genomics data or epigenetics data to interpret a GWAS result. And for single cell analysis this year, we are going to increase this to 3 lectures, 2 on single cell RNA-seq analysis and 1 on single cell ATAC-seq for understanding gene regulation. And the last module is going to be on cancer genomics and cancer immunology. We have a pretty heavy coverage on cancer, partly because I work at Dana-Farber Cancer Institute, but also because right now in genomic technology and computational algorithm development, cancer field really benefits from the cutting edge of these new development. All the newest techniques, all the newest computational methods are being applied in cancer genomics, functional genomics, and precision medicine and cancer immunology. That's why we are gonna have a pretty heavy coverage. We're gonna teach you about a US consortium to generate tumor profiles of over 10,000 tumors, how to look at subtypes of one cancer type, how to look at survival analysis. For precision medicine, we want to kind of show you how to analyze drug screens and genetic screens such as CRISPR screen data, and also look at tumor samples to understand how tumor evade the human immune system, and how immunotherapy is really helping to cure cancer. And so that's the last module. So the course website is available from this link, but this is only available if you are within Harvard. We will have the earlier years of lecture videos available. I apologize, actually slides are no longer available because I think Canvas changed the link location and they removed the files. And so I will try to make the slides available before the lecture and post them onto the Canvas website. There have been video tutorials created previously from earlier years of students, and so we will have a link there. You can also look at the links to the previous labs. We will also record this year's lectures and labs. For discussions, we are not going to use Canvas because we realize there are students who might be interested in taking this course from anywhere else in this world. They may not have access to to Canvas, but they might still want to join some discussion. So we're going to have the course discussion on Slack from this link. I'll also put this link on the course Canvas website. So if you want to take the course remotely, or if you have a time conflict with this course, it's okay. We're going to record all the lectures and put them on the Canvas website. And this year, I also will create a Git book. And this link is already available. We have a prototype textbook, which is currently linked to previous years of lectures, and we will gradually be updating them. And because this year the whole course is on Zoom, instead of giving many bad lectures throughout the semester, I'm only going to give maybe a third or half of the lectures to live to be updated. Some of these lectures can also be given by my colleagues, not from Harvard, from elsewhere in the US, who are better expert in this area than me. And so you can really get the best from all the faculty in this field. But we will keep some of the old lectures there. I am aiming to improve this course gradually over the years, so we will only update maybe a third or half of the lectures every year. And so on the days that we will not have live lecture, I will post the old lecture videos online ahead of time and send an email announcement. So students will know that that day we will not have a Zoom lecture, and you can just watch the previous years of videos. And so just watch for your Canvas email announcement. This course will have 6 homework. You will submit the homework on Canvas. This is the Harvard website. And for each homework you are going to submit, make sure you include a R Markdown and also the HTML output. Write down your name and the course that you are taking, whether it's Stat 115, 215, or Bio 282, and which homework it is. So out of the 6 homework, homework 1 will be 9 points. This is the same for undergraduate and graduate students. I would expect this homework to take roughly 10 hours on average. If this is taking you way more time than this, you might consider whether to get better prepared for taking it in future years. Homework 2 to 6 are going to be more material. We expect it to have about twice as much material, but it will count 15 points for the undergraduate course and 20 points toward the graduate course. We will distribute just one set of problem set for each homework, but on questions that are labeled for graduate student only, that means that if you are only taking Stat 115, you don't have to do those. But students who are taking the graduate level of the course, you have to do all the questions in that problem set. And that's why graduate course have more points. At the end of the course, we will curve the undergraduate students and the graduate students, the students taking the 200 level courses separately, because your homework are slightly different. But we'll make sure that the undergraduates will kind of cover a subset of the graduate material. That said, if you are taking Stat 115 and you want to really try it out to answer all the questions in the homework, you're welcome to do that, and we can give you some bonus points there as well. After each homework, there is going to be an online survey directly on Canvas, and we hope all the students give us feedback on how well you feel the lecture is running, the labs are running, and how many hours did you spend on homework. There are some questions that are too hard or too vague or took you too much time to do, we would really love to hear from you on that, which will help us improve this course for future years. And so as a small incentive, we give students 0.2 towards your final course credit. And so these are not added up to 100, but we will scale it to 100 points at the end. The homework survey is 0.2 points each. At the very end of the year, we're gonna have an optional credit, about 5 points, to ask students to help us design a new question for next year's homework. This way we will have some new questions for students. It's not an easy thing to do. I'm hoping that later on you will see from this doing the homework, we try to use some exemplary data to show you how to use different algorithms to touch on all the topics we covered in the lecture. So you can see how the different material fits together into each other. In order to create this homework, it's actually a lot of effort. And so unfortunately, we cannot update all the questions every year. It just takes too much effort, but we're hoping that through this, you can see how it's done, which means if you want to do the homework, we encourage students to discuss together, but you have to turn in your individual homework. You cannot turn in team homework. You need to submit your individual one, but you can discuss these questions on the Slack. There are a lot of trial and errors involved, and we really hope students can help each other. So as we mentioned, homework 1 is released today, and it's going to be due in 2 weeks' time. All the remaining homeworks, usually it's spaced at 2 weeks apart. This year, one thing is different. Normally we have a whole week in March of spring break, but this year there are 5 different wellness days. And so in those days, there are no lectures. And so the first homework is due on Sunday midnight. But I think as we see more and more wellness days, some of the homework might be due on Tuesday or Wednesday or Thursday. But we will give students 6 late days throughout the semester. You can choose to use 1 day on each homework, or you can choose to use 2 late days on the last 3 homework or whatever. But you have 6 late days throughout. We ask that you don't take more than 3 days on 1 homework, because we have to leave time for the TA to grade the homework. By the way, if you take additional late days, after you use the 6 days, each additional late days will incur 10% daily penalty. And we really don't encourage students to wait too long. After you use, say, 3 days on a homework, 3 late days on homework, it's time to move on. Submit whatever you have, because the next homework is coming up very quickly. And so it's time to kind of work on the new questions. For labs, we have regular labs where the TA will really show you, like command line, you can see the screen, or, show you how to run something, how to do some specific type of coding on Tuesday at 6 p.m. and also Thursday at 8 p.m. These will also be recorded. And if the Tuesday recording is done very well, the TA will post a tutorial video on the website as well. We will also make those available on the Gitbook. If the recording is not done very well, the TA might record again on Thursday. But if it's recorded well, they will send an announcement. So Thursday will just be student asking questions. And on the weekends, we will have office hours. Those will not be kind of lecture style labs. You can watch the video recording. That's mostly kind of like an office hour to help students with homework questions. So the labs, well, as we mentioned, the lecture style lab will be recorded as well. And the Zoom information is also on Canvas, but the recorded videos will be put on YouTube and be available on both Canvas and the Gitbook. One important thing on the Canvas is that we need all registered students to sign up for a Canon account. This is the big Harvard cluster. You will need a Canon account to do the homeworks. So please sign up today so we can create the Canon account for you as soon as possible. And in the first lab, the TA will teach you how to log in, how to move around between directories, how to submit jobs, especially if some of the jobs are time consuming, how to run them in the queue. And so lab will start this week, starting Tuesday tonight. And so make sure that you get your Canon account so that you can go to the lab and start learning right away. And so the labs are going to be super, super useful for homework. They are going to teach you how to run a lot of the software, how to do a lot of the coding, and the TAs will help you answer questions. We will try to, we understand that this lecture cell lab are only available in the evenings because during the day, the TAs also have to take their respective classes. But on the weekends, when we have office hours, we will try to have office hours in the morning as well, in case you are in a different time zone. Okay, I think that's all the information for the course, and I hope you will decide to take the course and enjoy learning. It's going to be a lot of work, but I hope you will find it really rewarding. There are, I hope that you will come back to this course later when your research topic is on this area. You watch the video, look at the homework, and really learn more material from the same course and hopefully understand even more. Okay, so good luck.